pull, open, be fine. And voltage gates for sodium open up at minus 55, 55. So you don't have to go from minus 90 to minus 50 like you do in a muscle cell here. Thank you. Yes.
Just across the hall, right?
Just across the hall.
Okay, let's go ahead and get started here. We're going to take a look at, we're continuing our discussion on the nervous system, and we're going to pick up where we left off the other day. If you have any questions on anything that we're talking about, this is a great time to raise them. I'll be more than happy to try and answer them for you. If you have any questions on anything as we go along, let me know, and I'll be glad to answer the question. So, okay, let's, can you post my grade on lab exam street? Okay. Yeah, it should have been on there. I don't know why it wasn't showing up, but I'll go back and look at it. It should be there because I mean, I can see it on my end for the lab exam. I don't know why it's not showing up where you can see it. But I'll go back and see. There's probably something that wasn't clicked right on my end, that's why it's not there. This isn't the first time stuff like that's happened. So let me go ahead. I'll take care of that for you as soon as we get out of class. So, any other questions? Okay, then I'm going to go into. There we go. Okay, we were talking the other day about membrane potentials. About membrane potentials in the nervous system. A neuron is got a charge on it. Its resting membrane potential is about -70 millivolts. Different from skeletal muscle, which is around -90. So we're at -70. Threshold in a neuron is about -55. So the amount of depolarization it takes to generate an action potential in a neuron is only 15 millivolts. We only have to go from -70 to -55, and then we can get an action potential running. Still using sodium and potassium, but we'll get, it takes a lot less change in the charge to get us to threshold. So let's see what this has to say for us.
The human brain alone contains about 100 billion nerve cells called neurons. A neuron, like all cells, has negatively and positively charged ions in the fluids located both inside and outside of the cell. However, the ionic composition of these two fluids is markedly different. The inner surface of the membrane is negatively charged, while the outer surface is positively charged. This unequal distribution of charge creates a voltage across the plasma membrane, known as the membrane potential. In a resting, unstimulated neuron, this membrane potential is known specifically as the resting membrane potential. The value of the resting membrane potential depends on two factors. The first is the presence of concentration gradients across the plasma membrane, especially those gradients for sodium and potassium ions. The second is the permeability of the plasma membrane to sodium and potassium. In the resting state, this permeability depends on the number of non-gated leak channels present in the membrane for these ions. The magnitude of the resting membrane potential, inside relative to outside, is measured in millivolts using a voltmeter and 2 electrodes. We'll now place both electrodes in the extracellular fluid. Note that the voltmeter records 0 voltage. This is because the unequal distribution of charge exists only across the plasma membrane. The intra and extracellular fluids are otherwise electrically neutral. However, when we insert one of the electrodes into the intracellular fluid, a voltage is recorded. The value of the resting membrane potential varies, but on average, it is -70 millivolts.
So the charge on a membrane of a neuron is -70 millivolts. It still has to do with sodium concentrations and potassium concentrations. Sodium is higher outside than it is inside. Potassium is higher inside than outside. The outside of a neuron is usually positively charged, just like a muscle cell, and the inside is usually negatively charged, again, just like the muscle cell. Yeah. there are changes that occur in the membrane potential. They occur all the time. Now, the changes occur when we see ions moving across the membrane. Now, how do we get the ions to go across the membrane? Well, despite the fact that there are leakage channels that allow sodium to come in and potassium to go out, and we regulate that with the sodium potassium pump so we don't have an excessive amount of sodium inside or potassium outside. We don't want that. The charge on the membrane changes drastically when we open up or close the voltage gates or the chemical gates or the mechanical gates to allow sodium to come in and potassium to go out. And we have large quantities moving across the membranes then. The permeability of the membrane, meaning the ability of ions to pass through the membrane, increases drastically or it stops drastically. Now, there are two types of changes that occur. Two types of changes of the charges. We have our well-known action potential, just like we have an action potential in muscle cells. The action potential here is going to travel down the axon. But we have another type of potential here called a graded potential. Graded potentials are something new to us, at least in this class. Graded potentials are short-term, short-lived potentials that actually depend on the strength of the stimulus. Action potentials don't care about the strength of a stimulus. If you hit threshold, the action potential is going to run. It doesn't matter how strong the stimulus was, whether you were smashing your thumb with a hammer or tapping your thumb with a pencil or a nail or whatever. If you hit threshold, you're going to generate an action potential. And the action potential isn't going to care if it was caused by a hammer or a pencil. They're all going to run. Every action potential looks just like all the others. Graded potentials, however, deal with intensity. The stronger the stimulus, the greater the stimulus, the longer the graded potential can last. The graded potential will wear out. It will run and it will wear out before it gets somewhere, or it may reach different locations in the neuron. It just depends. The graded potential is what we use to respond to a stimulus. The graded potential will be the trigger that will give us an action potential. The graded potential is what we use to get to threshold in the neuron. If the graded potential isn't strong enough to get to threshold, to depolarize the neuron to threshold, nothing happens. We aren't aware of the stimulus. There are lots of stimuli that occur to our bodies that we're not aware of because the graded potential generated by the stimulus didn't hit threshold. So we didn't know about it. And that's not necessarily a bad thing, because if we didn't filter out many of these stimuli, we would be overwhelmed. So graded potentials and action potentials. Graded potentials can lead to action potentials. Graded potentials can lead to action potentials. A graded potential will occur when we open an ion channel. In this case, let's say that sodium is rushing in. Okay. And a graded potential is a localized depolarization event. It's a localized depolarization event. Sodium comes in, and we're changing the charge on the membrane on a dendrite. That's where the sensory receptors are usually located. We spread a depolarization wave spreads across the dendrite and may head down towards the cell body of the neuron, or it may just run out. it may not be strong enough to stimulate a depolarization wave across the membrane of the dendrite down into the neuron. And it can be, the channel can be a ligand-gated channel. Be a ligand-gated channel, like a neurotransmitter lands on the dendrite and opens up sodium channels, but maybe it doesn't open up very many. It can be a mechanoreceptor, a mechanical gate. You've deformed the end of the dendrite. You've touched something. It opens up sodium channels and sodium comes in and you depolarize the end of the dendrite. But did you depolarize it enough? In the case of the graded potential, the strength of the signal really comes into play. If it's not strong enough to cause a depolarization of the dendrite, which is the receiving end of the neuron, if it's not strong enough to depolarize the end of the dendrite to where we get a depolarization wave that hits threshold in the cell body of the neuron, nothing happens. Graded potentials will start in the dendrites. They'll start on the cell bodies. Action potentials, however, are different. They will only trigger in an area called the axon hillock. The axon hillock is where the axon leaves the cell body. That's the only place that action potentials are generated. That's where we hit threshold. That's where a depolarization wave coming from the dendrite or coming from the cell body itself has to hit threshold. Action potentials are only triggered by voltage-gated channels. When you hit threshold, you change the charge in the membrane from -70 to -55. That opens up voltage gates for sodium. The only place we have our voltage gates for sodium are down by the axon hillock. We hit -55, those gates open, sodium rushes in, we depolarize, we have an action potential, and it's off and running. Graded potentials are only triggered by ligand gates or mechanical gates. And again, the graded potentials will spread, but they will wear out. Action potentials will run until they're done. They'll run the entire length of the axon, no matter how long it is, and no matter how many axon terminals there are. If there are 10,000 axon terminals, the action potential will go to all 10,000 axon terminals. Doesn't matter. Doesn't matter if the axon's 3 feet long or 3 inches long. Every terminal will receive the same action potential, and it won't wear out. How do we get a graded potential? Well, some sort of mechanical stress. We distort a mechanical gate. We put pressure on it. We press on something, or something presses on our skin. We pinch our skin. We hit a pain receptor. We deform the end of a dendrite. And sodium comes in because we've opened up the mechanical gates. Or we have a neurotransmitter gated channel. Perhaps acetylcholine lands on the sodium gate and opens it up. Okay, that's great. Sodium comes in and we're having a localized depolarization. Did we have enough neurotransmitter? Was there enough? Did we get enough sodium gates opened up? So those are the two sources, mechanical distortion or the use of a neurotransmitter. And so depending on the strength of the stimulus, we may or may not open up enough gated channels. Strength of the stimulus determines how many gated channels are going to open up, and that determines the strength of the depolarization wave, the localized depolarization wave spreading down the dendrite towards the cell body. It's a localized depolarization event. That's all it is. And you may or may not get the threshold at the axon hillock. So because our graded potential wears out as it spreads away from the initial stimulus site. The charge in the membrane, we depolarize initially, but it's going to gradually decrease. The key is the charge sufficiently strong enough to get the threshold at the cell body? Or nothing happens. You know, we're not aware of the odor, we're not aware of the sound, we're not aware of the touch, the pressure, the pain, the temperature. We don't detect it. We don't perceive it. On the other hand, an action potential is going to be constant. It's all or nothing. If we hit threshold, we have an action potential. It will run till it's done. Graded potentials are variable. Now, we do know that and also we know that an action potential, the action potential is not dependent on the strength of the stimulus. Once we hit threshold, we get an action potential. Doesn't matter how strong the stimulus was. Again, if you got smacked in the thumb with a hammer, the action potential for that is not going to be any different than the action potential from tapping your pencil on your thumb. What we will notice is the frequency of occurrence of the action potentials will go up. The intensity of the stimulus shows up in the number of action potentials per second that we're generating. The frequency of occurrence. A very powerful stimulus will give you lots and lots and lots of action potentials. A weak stimulus that still hit threshold will just give you a few action potentials. So we can still determine the intensity of the stimulus by how many action potentials we get. But the action potentials are all going to be the same size. And the action potential will run all the way through. You know, once the action potential has formed at the axon hillock, it will run all the way to the end of the axon and will not wear out. Now, you know, size matters a little bit here. The size of the axon determines how rapidly we can get the action potential down the nerve. Large axons, large nerve fibers that are myelinated send signals faster than the unmyelinated axons. But that's the only difference here. Now, graded potentials, they stop as soon as the stimulus stops. The action potential will run until it's done. So the stimulus may have stopped. The action potential is still going to keep on running. Because it's going to run from the axon hillock all the way to the last axon terminal, however far away that is, how many of them there are. The graded potential, as soon as the stimulus stops, it stops. Graded potentials have no refractory period either. That's a period of resetting everything, resetting the gates. We don't have that with the graded potential. We have a refractory period in the action potential. We have to reset our voltage gates in the action potential. That takes a little bit of time. And so every action potential that's generated has a refractory period that has to occur before we get the next action potential, so that the voltage gates have been reset, ready to go. So, okay, action potentials. This is how we signal through muscles and through nerves, through neurons. Only in muscles that we get action potentials spreading in muscle cells. They're delivered from a motor nerve. We've just talked about that in muscles. And the action potential spreads all the way through the muscle cell, so it can trigger the opening of the sarcoplasmic reticulum to release calcium to bind to troponin and roll the tropomyosin out of the way so the myosin heads can attach and we can get a contraction. That's why we have action potentials in muscle cells. In neurons, it's communication between neuron to neuron, neuron to muscle, neuron to gland. The action potential is a change in the charge on the membrane. We generally see it going from -70 up to +30 and back to -70 with a brief undershoot to -90. So it's about 100 millivolt change in the charge, generally from -70 to +30 and back to -70. We often call the action potential a nerve impulse. Okay, we'll call it a nerve impulse, but it's really an action potential. And it is always voltage gates. It's always going to be dealing with voltage gates here. Yeah.
Neurons communicate over long distances by generating and sending electrical signals called action potentials. Most action potentials originate in the axon's initial segment, a region also called the trigger zone.
Or axon hillock.
During an action potential, voltage-gated sodium and potassium channels open and close, thus altering the membrane's permeability to sodium and potassium ions. As these ions diffuse through the channels, the membrane potential changes. It depolarizes from the resting state of -70 millivolts to a peak value of +30 millivolts, and then quickly repolarizes back to -70 millivolts after a brief period of hyperpolarization. In this topic, we explore, step by step, the events that occur as an action potential is generated in the axon's initial segment.
Okay, so we have depolarization, repolarization, and hyperpolarization. Hyperpolarization is that undershoot where we go all the way down to -90 millivolts and then come back up to -70. And this gives us the time we need to make sure that the voltage gates for sodium have been reset. So 4 steps in an action potential. In the resting stage, the charge on the membrane is -70 millivolts. The sodium and potassium channels, the gated channels are closed. So nothing's going to open, they're not open. When we depolarize the membrane, we open up the sodium channels. Depolarization involves the sodium channels. You know that from muscles. So we have a graded potential that with its localized depolarization event spreading across the neuron cell body. We are opening up the sodium gated channels here because it's a localized depolarization, and we hit threshold of -55. We only have to go 15 millivolts to go from 70 to 55. And so we hit threshold and the voltage gated sodium channels open up. Sodium rushes in, changes the charge on the membrane from -55 to +30. Yeah, because we started out at -70, the graded potential depolarizes it to hit threshold at -55, and we go all the way up to +30. At that point, we start repolarizing. At +30, just like in muscles, at +30, the sodium gates shut and the potassium gates open, and sodium and potassium starts leaving the cell. And we see the charge on the membrane starts dropping from +30 all the way down to -90. We are repolarizing. We don't care that the ions are different now. We don't care that we end up now at repolarizing, we got potassium outside and sodium inside. What we care about is the charge. We want to put positive charges back on the outside of the neuron. Because remember, when we started out, the neuron was positive on the outside and negative on the inside. Now we've got all this sodium rushing in, changing the charge to where we're positive on the inside. So by opening up the potassium gates, potassium leaves, reestablishes the charge of positive on the inside and negative on the outside. Now we'll correct the ion concentrations with the sodium potassium pump. All we're concerned about is putting plus on the outside and minus on the inside. So we're repolarizing and we go all the way back down to minus 90. Repolarizing, we are polarizing the membrane to the charge it had before. Positive outside, negative inside, the polarity we had before. When we undershoot past minus 70, we are hyperpolarizing. The potassium channels stay open. Some of them stay open, and this gives all of our voltage sodium channels time to reset. And we allow, by having the potassium channels open, we still have more potassium leaving the cell, and we end up making we keep the inside negative, and we allow the sodium channels to continue to reset. And then once we have reestablished that the sodium channels are all reset now, because it takes some time, then we start recovering the sodium and we come back up to minus 70 and we are ready. Sodium potassium pumps go to work. We reestablish the ionic concentrations, sodium outside, potassium inside. It's all done in milliseconds. We're not aware of it. The repolarization reestablishes the charge. Repolarization puts the positive charge on the outside and the negative charge on the inside. Doesn't matter that it's potassium outside and sodium inside now. We'll fix that in a millisecond with the sodium potassium pumps. What we wanted to do was get the charge back to where it was with a positive charge outside and a negative charge inside. Doesn't matter that we're using potassium and sodium in a different role now. We'll very quickly fix that. So, okay. Okay, so action potentials. You know what depolarization is. Depolarization is the influx of sodium. Depolarization is the outflow of potassium. An action potential is an all or none phenomena. It either goes or it doesn't, and if it goes, it will go all the way to the end of the axon. It won't stop in between, it won't wear out. It is just as strong at the end of the axon as it was at the axon hillock. When we hit threshold, the action potential will run. Threshold is minus 55. It's a very low threshold. You know, we don't have to go from minus 90 to minus 50 like we do in a muscle cell. We only have to go from minus 70 to minus 55. So it's a very easy threshold to reach. We hit threshold, the sodium gates open, sodium rushes in. We hit plus 30. You see in the graph there, we go all the way up to plus 30. Sodium gates shut, potassium gates open, and there's depolarization. We get all the way up to plus 30. And then the sodium gates shut and we repolarize, but we will dip. The repolarization will take us into a hyperpolarized state where we dip down to minus 90 and then come back up to minus 70. It's only when we get down to minus 90 that the potassium gates close, and then we reestablish our charge of minus 70. So here we have a depolarization, repolarization, hyperpolarization event. We're not going to get an action potential from every depolarization event. Not every stimulus is going to trigger an action potential. You've got to hit threshold. If we don't hit threshold, nothing happens. And there are many stimuli that we don't respond to because we're not aware of it. If we don't hit threshold, then we're not aware of the stimulus. And as I said, that's a good thing, because otherwise we'd be overwhelmed by all the stimuli going on around us. We're filtering out the stimuli that we don't need to hear. You know, the stimulus has to be raised to our level of awareness. So, and we're not going to perceive it unless it can hit threshold. So an action potential either happens completely or it doesn't happen at all. We have to propagate the action potential all the way down the axon, from the axon hillock to the axon terminal. And just like we have in a muscle, the sodium influx on a voltage gate on the axon will depolarize that region and open up sodium gates next to it. And they depolarize. And then the ones next to that depolarize. So we proceed essentially in one direction only down the axon. As we go down, we depolarize and then we repolarize behind it. As soon as we depolarize to plus 30, the sodium gates shut, potassium gates open, and we start the repolarization. So it's a wave of depolarization followed by repolarization for each segment of the axon that is stimulated.
Action potentials propagate in a continuous fashion in unmyelinated axons. Once an action potential is generated in the initial segment of the axon, it propagates the entire length of the axon. Recall that a threshold stimulus causes voltage-gated sodium channels to open. The influx of sodium ions generates an action potential. It also establishes a depolarizing current that flows to the next segment and brings it to threshold. Voltage-gated sodium channels open, regenerating the action potential in this segment of the axon. Current flows from this segment and depolarizes the next segment to threshold, thus regenerating the action potential yet again. In this way, regeneration continues in one direction all the way down to the axon terminals. The basis for unidirectional propagation is revealed when we take a closer look. By the end of the depolarization phase of the action potential, all voltage-gated sodium channels inactivate and voltage-gated potassium channels open. These two events render this segment of the axon temporarily insensitive or refractory to another depolarizing stimulus. However, voltage-gated sodium channels in the downstream segment are closed and receptive to a depolarizing stimulus. Thus, propagation occurs sequentially down the axon to the axon terminals. In myelinated axons, action potential propagation is a bit different. Here they propagate in a saltatory or leaping fashion. The myelin sheath consists of multiple layers of tightly wrapped glial cell membrane. But this sheath is not a continuous one. Exposed areas of axonal membrane, known as nodes of Ranvier, occur at discrete intervals. Voltage-gated sodium channels are abundant in the nodes, but largely absent between nodes. So action potentials are regenerated at each node, not in areas covered by the myelin sheath. However, the myelin sheath does provide the insulation necessary for the rapid spread of depolarizing current. And the sooner the nodes reach threshold, the faster action potentials propagate along the axon. Saltatory conduction is extremely fast. Velocities often exceed 100 meters per second. In contrast, continuous conduction is fairly slow. Velocities rarely exceed 2 meters per second. Nevertheless, both continuous and saltatory conduction propagate action potentials over varying distances because action potentials regenerate along the way. Summary. Propagation of an action potential. Once generated, the action potential propagates the entire length of the axon without decrement.
Okay, so we saw a little bit there on saltatory conduction, which is the method we use to jump from node to node on the insulated myelinated axons. More about that in a little bit here. So we know that action potentials take place in the axons only. They start at the axon hillock. The speed of propagation depends on a couple things. The action potentials are always going to be the same. The larger the size of the axon, the faster you're going to have the propagation, simply because it's like looking at a hose. A garden hose is going to send water through at 5 liters a second, but a fire hose, a 3-inch fire hose, for example, may send water through at 30 liters per second. Bigger is better. So because we have less resistance. The smaller diameter axons have greater resistance than the larger diameter axons. That's one way we increase the speed of transmission. The other one is dealing with the myelination on the axon. If you have myelin on your axon, if your axon is myelinated, it will use a process known as saltatory conduction, where we are jumping. Salta means to leap. It's Latin for leap. We are jumping from node to node. We bypass the intermediate areas, and so our action potential jumps from node to node and can travel extraordinarily fast in getting to the axon terminals. Continuous conduction requires us to depolarize and repolarize every segment of the axon. And while 2 meters per second sounds slow, remember that the areas that are not myelinated are going to be axons that are very, very short, less than a millimeter in size. So while 2 meters per second is not all that fast, when you're dealing with millimeters, it's fast enough. Okay, continuous conduction is slow. Saltatory conduction is 30 times faster than continuous conduction. The myelin sheath insulates the axon. The myelin, the lipoprotein, is wrapped around the axon. It functions like insulation on a wire, and it allows us to send a much faster signal. Now, the action potentials, again, are all the same size, but they can travel faster down the myelinated axon. And we simply jump. The action potential jumps from node of Ranvier to node of Ranvier, from gap to gap. So here we see continuous conduction versus saltatory conduction. With saltatory conduction at the top, the signal has to pass through the entire segment of the neuron down to any axon terminals that are present. But with saltatory conduction, we jump node to node to node. Problems. There is an autoimmune disorder known as multiple sclerosis. It attacks the myelin sheath. It, for whatever reason, we don't, like most autoimmune disorders, we don't know what's going on. We don't know why it does what it does. But the myelin sheath is attacked by our own immune system, and it becomes hardened. It becomes what we call a sclerotic lesion. A lesion is a sore or an infected area. Well, here we have a hardened lesion called a sclerosis, and that breaks down the ability to conduct action potentials. And so once we start breaking them down, we stop sending them. And so the axons become unmyelinated again. They lose their myelin. And so the signals that would be coming down the axon are disrupted. It's like scraping the insulation off of a wire and expecting it to still work. Now, in this case, multiple sclerosis will affect axons in different organ systems at different times. Like most autoimmune disorders, it comes and goes. We say it waxes and wanes. When it's present and when it's waxing, it can affect the digestive system or the skeletal muscle system. or the or equilibrium or vision. And so, and then it'll go away and it'll come back, and it may come back in a different system. You know, it can cause problems with vision. It can cause problems with muscles. It can cause problems in speaking, controlling your bowel and bladder, overall muscle weakness, and then it'll go away and it'll come back. It may go in remission for years and then come back in a different system that it's going to affect, because it's going after the axons, not the, you know, it's going after the individual axons that innervate a system, not the organs themselves. So, and then it'll go away. Now, how do you treat it? Well, you know, immunosuppressants, which opens up a whole other area of concern. So we treat it with immunosuppressants, because like any autoimmune disorder, we don't know what caused it, and we don't have a cure for it. So now it's possible. Now here's something that's more recent information. It's possible that high levels of vitamin D may prevent its onset. Don't know. Yeah. That's a possible type of prophylactic medication there, but high levels of vitamin D may prevent it from developing. So, okay. Something else we see with impairing our action potential transmission. You ever had dental work done? You know, and you go into the dentist's office and you get in the chair and they say, okay, I'm going to numb you up now, and they give you a couple of injections of Novocaine or lidocaine or something like that. Or maybe you've had surgery, you know, maybe you've had outpatient surgery and, you know, on your finger or your hand or something, and you're awake for it. Or maybe you've had arthroscopic surgery, and what is used to block the pain signals is some local anesthetic. Novocaine is a great choice as a local anesthetic. What Novocaine does is blocks the sodium gated voltage channel, the sodium gated voltage channels. Yes, that's the phrase I was looking for. So we can't depolarize. Pain receptors are still being stimulated. Pain receptors are not shut down. All we do with Novocaine and lidocaine and any other pain medication like that is block the signal by shutting down the sodium channels. Sodium gates can't open, so they can't depolarize and send a signal up to the brain and say, hey, that really hurts. You ought to stop that. The nerve impulses can't get past the area where the blockage is. Now, when we have dental work done, usually the Novocaine gets injected into one of the branches of the trigeminal nerve, either for the upper, for the maxilla or the mandible. And so we don't feel something. It's still there. The whole side of our face is numb because not only are our pain signals being blocked, but everything else is too. Touch and pressure and temperature and itch. All those are blocked also. So we're blocking all the sodium channels. That's what lidocaine and Novocaine do. They block those channels. It's numb. Now, something else we do, you ever sleep on your hand? You ever fall asleep on your hand? You know, and you wake up in the middle of the night, your hand's numb, or you're sitting awkwardly and you get up and say, oh, my foot's asleep, because it's all numb. What you've done is you shut off the circulation briefly to that area of the body. And so without continuous blood flow, I mean, neurons are very sensitive to oxygen levels. When you reduce the oxygen level in the blood because you're impairing the blood flow, the neurons can't function well. And so your leg's numb or your hand's numb. You don't feel anything. On a cold day, your face gets numb. because the vessels supplying your face vasoconstrict, blood flow is directed away from your skin, and you no longer feel your skin because of this. The receptors, the sodium channels for the receptors are no longer functioning because we don't have enough blood supply delivering enough oxygen to those pain receptors or temperature receptors or whatever. Our foot goes to sleep, our face gets numb, our fingers get numb, your arms numb because you've been sleeping on it. That's a very routine phenomena that occurs. All because you are blocking the action potentials from getting to the brain. So now, how do though, I alluded to this earlier, how do we tell the strength of a stimulus? How do we tell the intensity of the stimulus? How do we tell the difference between a gentle touch and a more forceful touch? Remember that action potentials are always alike. Every action potential looks exactly like any other action potential. There's no difference between them. An action potential from a taste bud is going to be the same as look just like an action potential from your optic nerve. They're going to be the same. They don't care the intensity of the stimulus. An action potential from a hammer hitting your thumb is going to look just like the action potential from tapping your pencil on your thumb. You've got an action potential, yay, it looks the same. The only way we can tell the difference between a weak stimulus and a strong stimulus is by the frequency of the action potentials. If we have more action potentials per second, that tells us that then if we are seeing how we're not counting the number of action potentials we get per second, but the frequency of occurrence of the action potential. How many action potentials do we get per second? That tells us the intensity of the stimulus. The more action potentials we get, the more intense the stimulus is. A firm pressure stimulus gives us more action potentials per second. A firm stimulus, a powerful stimulus gives us more action potentials per second than a gentle, light touch stimulus. So we're going to get more per second. It's the only way we can tell about the stimulus intensity, because the individual action potentials can't do a thing. But we've got to have time between action potentials. They have to have time to reset the gates. An action potential fires. It runs down the axon. The axon has to be ready for the next one. It has to reset all of those sodium gates and potassium gates. If the sodium gates are open, we can't get another stimulus. Now we know that the sodium gates shut at +30. How long does it take them to shut down? We have to have them reset themselves. So we hit threshold at -55, all the sodium gates fling open. At +30, the sodium gates start to close. It's not quite as rapid. We have something here called the absolute refractory period, where the absolute refractory period is the time from the hitting threshold at -55 until we get to +30 and all the sodium gates reset. Because sodium gates have to close properly and be ready to go again when we hit -55. They close at +30. But the charge in the membrane, remember they close at +30, but the charge in the membrane is still high. We've got to get our membrane charge back down to threshold before they can go again. Because The gate closes at plus 30, but the charge in the membrane is still, is plus 30. We got to get the gates to close at plus 30, and then the charge in the membrane has to drop. And that's when we have to that's the resetting phase. We want to make sure that action potentials always occur when the threshold at minus 55 is reached. So we open the gate, it runs to plus 30, the sodium gates shut, and we're gradually repolarizing the charge in the membrane. We get down to minus 55. The sodium gates have reset. This is called the absolute refractory period. The absolute refractory period guarantees that that our action potentials are always going to be once and done every time. It's an all or none event. It also means that action potentials only go one way. We can't generate another action potential like we do in tetany in muscles. We can't do that. We can't get another stimulus in there until the axon is ready to go. So now after the absolute refractory period, we have something known as the relative refractory period. In the relative refractory period, most of our sodium gates have hit threshold and have reset. Some are still getting there and resetting themselves. Most of our sodium gates have gotten back to threshold. We are still repolarizing though. We still have potassium gates open, potassium still leaving the cell. We're still repolarizing, as I said. Our threshold is higher. Now, threshold is elevated. What does that mean? Well, the charge in the membrane, remember, we didn't go to minus 70 when we're repolarizing, we go all the way to minus 90. So a stimulus here to generate an action potential in the relative refractory period doesn't have to go from minus 70 to 55, it has to go from minus 90 to 55. And here is where a strong stimulus, only a stimulus that is strong enough to cause a localized depolarization that can go not from minus 70 to 55, but minus 90 to 55, will get that sodium gate to open up. So here we see, there's our neuron up there. We see sodium rushing in, we depolarize, we hit plus 30, and then we start to repolarize there. The sodium gates are going to be closing, and they're not going to be reset until we hit threshold again of minus 55. But the membrane is going to continue to become hyperpolarized. Now, this blue box outlines the area of the absolute refractory period. From the time we hit threshold to the time the sodium gates are reset is the absolute refractory period. Minus 55. We go from minus 55 to plus 30, and from plus 30 back down to minus 55. That is the absolute refractory period. We can't get another action potential generated there because the sodium gates are opening and depolarizing all the way up to plus 30. At plus 30, they start shutting and resetting themselves. But remember, the charge in the membrane doesn't automatically immediately drop to minus 55. Our membrane charge, when sodium gates shut, our membrane charge is still plus 30. We got to get the membrane to be repolarized back down to minus 55. Nothing can happen in this time frame in here. But once we get to threshold, then we go into what's known as the relative refractory period. If we have, notice that in the relative refractory period, the membrane has gone all the way down to minus 90. A stimulus can trigger an action potential here if, and only if, it's strong enough to depolarize not from minus 70 to 55, but from minus 90 to 55. it's got to be a really powerful stimulus to get an action potential here. Has to be a very powerful stimulus to get us to an action potential at this point. So we can get an action potential here, but because we have most of our sodium gates have been reset. Not all of them, but most of them. But again, the problem is that the charge in the membrane is now minus 90. Why do we have the charge like that? To make sure that all the sodium gates have been reset, all the potassium gates have been reset, so that we can do it again. Now, this doesn't take very long. We're not aware of it. We are not aware of the difference, the time difference here. But in the absolute refractory period, we cannot get another action potential generated. Only in the relative refractory period could we get a second action potential, but it has to be a very powerful stimulus to do that. Now, the action potential that's generated, though, will look just like all the others. It doesn't care that the stimulus was more intense, but it will, it has to be a stronger stimulus to get from here back up the threshold. Okay, so in summary, resting potential is minus 70, threshold is minus 55. We depolarize from minus 70 to plus 30. We hit threshold, we're going to depolarize from minus 70 to plus 30, and we're going to repolarize back down from plus 30, ultimately to minus 70, with a undershoot when we hyperpolarize. We can't get a second action potential until we have reset most of the sodium gates. So that the absolute refractory period is a time from threshold to threshold. This area back here. Now, area in blue. Now, do we care? Well, we don't notice it. We don't notice. We don't notice that at all. So to us, it's instantaneous. You want a good analogy to the absolute refractory period? Consider flushing a toilet. You flush your toilet, there's the action potential. It's a different way of looking at flushing a toilet. You generate an action potential by flushing the toilet. Now, if you pull the handle a second time right after the first time, you flush the toilet, there goes the water, and you pull the handle again, what's going to happen? Absolutely nothing. The toilet is in its absolute refractory period. It can't flush again until the tank fills up with enough water to make it go. And once you get it partly filled up and you can pull the handle again, now you're in its relative refractory period. Ideally, you wait till it's filled up again. So, because we know that when you flush a toilet, you can't pull the handle a second time right after the first time and have it do anything. Okay. Now, the synapse is something we encounter here. We've already mentioned the synaptic cleft. The synapse is the connection between a neuron and a muscle cell, a neuron and another neuron, or a neuron and a gland. It's an indirect connection. The neurons are connected by these synapses. Now, the synapses don't touch. Remember that the synapses don't touch. The neuromuscular junction doesn't touch, we don't touch the muscle cell with the motor neuron axon terminal. They don't touch. There's a gap. There's a synaptic cleft. The synapse is the junction, the indirect junction. It can be neuron to neuron, neuron to muscle, neuron to gland. Now, a couple of terms we use here. We call them the presynaptic. We have, if we're going, let's just assume we're going neuron to neuron. We have what's known as the presynaptic neuron. That's the upstream neuron. The action potential is coming down the presynaptic neuron. It's flowing downhill. That's the upstream neuron. the action potential is flowing down the presynaptic neuron, heading towards the axon terminal at the synapse. At the synapse, we have the axon terminal, we have a synaptic cleft, and we have a dendrite on the other side, ready to receive the information. We're sending information down a neuron. We go from one neuron to the next neuron. The presynaptic neuron is the sender. The postsynaptic neuron is the receiver. It's on the presynaptic neuron is upstream. It's above the synaptic cleft. The postsynaptic is downstream. It is below the synaptic cleft. The axon terminal is located right at the synaptic cleft between it and the dendrite, and they don't touch. The postsynaptic neuron will receive the information. So, synapses can occur classically axon to dendrite, or rather axon terminal to dendrite. We see up there on the animation, we see the green axon and axon terminal, and we see the red action potential coming down. I don't think they're red. Coming down to the neuron, and we see the little yellow neurotransmitter getting released, and we see our action potential being repropagated in the postsynaptic neuron. The green structures there are the axon terminals from the presynaptic neuron, and the classic synapse is usually axon to dendrite. But we can also have axon to cell body. That's what's happening over here in the bottom area. It's okay. We can go axon to cell body. We bypass the dendrite. It still works. Sometimes we can go axon to axon. Pretty unusual. Dendrite to dendrite. Even more unusual. And sometimes we can go dendrite to cell body. That's very unusual, but it does happen. But usually it's going to be axon to dendrite or axon to cell body. They don't touch, but they allow the action potential to be repropagated on the downstream neuron. Now, the synapses can be either electrical or they can be chemical. An electrical synapse is pretty unusual. They usually involve gap junctions. So they usually involve holes in the cell membrane that are shared between, cells. Gap junctions are openings in the cell membrane that allow the cytoplasm of one cell to connect directly to the cytoplasm of the adjacent cell. We find this in eye movement. We find this in emotions. We find this in memory, so that we have very rapid movement, electrical movement from cell to cell without having to waste the time, if it's a waste, of the more classical depolarization, repolarization event where we connect directly like this. Eye movement requires rapid decision making. The hippocampus tells us where to store our memories. The hippocampus involves memories. We give emotional responses to memories, and that is rapid because we're using electrical synapses here. Most of the synapses, on the other hand, are going to be chemical and use neurotransmitters. The typical chemical synapse has an axon terminal on the presynaptic side, filled with synaptic vessels containing a neurotransmitter. The postsynaptic membrane, postsynaptic neuron, is usually going to be a dendrite, and it's going to have receptor sites for the neurotransmitter. Now, those receptors can either open or close chemical ligand gates. We don't have to see the neurotransmitter open up a gate. It can close a gate. It doesn't have to open sodium. It may open potassium. Depending on what the neurotransmitter is, it can either depolarize the downstream neuron or hyperpolarize it. So we use the classic approach of the chemical synapse with the axon terminal and the dendrite. The synaptic cleft is in between here, and we use the synapse. The synapse is used to slow down the transmission of the action potential slightly. It slows down the transmission to make certain that the postsynaptic neuron, the downstream neuron, is ready to receive a signal. That's why there's a time delay there. Not much of a time delay, but the action potential comes down, the presynaptic neuron reaches the axon terminals, neurotransmitter gets released across the synaptic cleft, it lands on the postsynaptic neuron, opens or closes gates. It may open a gate, it may close a gate. We don't know, typically we would think of acetylcholine opening a sodium gate, and we'll go with that for right now. There's a little bit of a delay to do that. And then we generate the action, we repropagate the action potential on the postsynaptic side. And then we're off and running till we get to the next neuron, or maybe the next to the gland or to the muscle cell. The reason we use the chemical synapse is that we don't have a hardwired connection between the presynaptic neuron and the postsynaptic neuron. Since it's not directly connected, the signal can only go one way. We don't want a signal going backwards. And so we prevent that by having this a one-way transmission. And it works. It works very well. The action potential comes down to the axon terminal of the presynaptic side. It releases the neurotransmitter. We have to have enough neurotransmitter to open up the gates on the postsynaptic side to repropagate the action potential, and then we're off and running again. Doesn't go backwards. It's a one-way movement. And we have to have enough neurotransmitter present to get it to work. So the neurotransmitter comes across the synaptic cleft and lands on specific and unique receptors on the postsynaptic side. It depends on a lot of things. It depends on the type of neurotransmitter. It depends on the type of receptors that are there. It can either be an excitatory or an inhibitory response, depending on what we're doing. In acetylcholine in skeletal muscle, it is an excitatory response. We release acetylcholine into skeletal muscle and it lands on the motor end plate and it opens up sodium channels and sodium rushes in and the skeletal muscle depolarizes the motor end plate and then hits threshold and we have an action potential. That's an excitatory response. But in the heart, acetylcholine is not excitatory. Acetylcholine is inhibitory. When we release acetylcholine into the heart, it slows the heart rate down. So our response can either be excitatory or inhibitory. We have 60 or so different neurotransmitters that can do different things. In some cases, they can either be excitatory or inhibitory depending on where they're being released. Acetylcholine is excitatory in skeletal muscle, but it's inhibitory in cardiac muscle. So in either case, though, when we land on the ligand gate, we will generate a graded potential. The graded potential will turn into an action potential that may do one of two things.
There is a narrow gap, the synaptic cleft, between the synaptic terminal of ascending neuron and the surface of a receiving cell. An action potential arrives at the end of the axon of the sending cell. This causes chemical changes that make vesicles containing a chemical called a neurotransmitter fused with the plasma membrane. The neurotransmitter molecules spill out into the synaptic cleft, diffuse across, and bind to the receptors on the receiving cell's membrane. The binding of neurotransmitters to receptors causes the attached ion channels to open. Depending on the type of neurotransmitter, receptors, and channels involved, this may excite or inhibit the receiving cell. Here we show excitation. Sodium ions rush in, changing the polarity of the membrane, and an action potential is triggered in the receiving cell. The neurotransmitter is broken down by an enzyme, and the ion channels close. Enzymatic breakdown is one of the several mechanisms by which transmission is terminated at chemical synapses.
Now, this is an excitatory response right here. What if we open up potassium channels and we hyperpolarize the postsynaptic neuron? That's an inhibitory response. We're driving the charge on the resting potential of the membrane from -70 to -90, making it harder to get to an action potential. So we may end up inhibiting the action of the postsynaptic neuron. It just depends. So here we see an example, one that we're very familiar with already. We've seen it before. This is the axon terminal with its calcium gate that's needed to release the neurotransmitter into the synaptic cleft of a skeletal muscle. And we have voltage-controlled calcium gates in the axon terminal, and that will, when those gates open, calcium comes into the axon terminal and allows the vesicles that contain neurotransmitter to fuse with the membrane of the axon terminal, releasing the neurotransmitter into the synaptic cleft. This is what we see taking place in our skeletal muscle cells. The time for this to take place is ½ millisecond. So here's what's going to happen. Sodium's coming in. We're having our action potential move down the motor neuron. The change in the charge opens up the calcium voltage channels. The neurotransmitter gets released into the postsynaptic side. The time it took for that was ½ millisecond. The time the action potential reached the axon terminal to the time the neurotransmitter showed up on the other side was ½ millisecond. And so now whatever the neurotransmitter is, it's on the other side now. It's on the postsynaptic side. Is it opening up sodium channels? Is it opening up potassium channels? Don't know. But this is, in our case, in our skeletal muscle, this opened up sodium channels and we got a depolarization event that may have led to an action potential. The delay, the synaptic delay, is anywhere from 0.3 of a millisecond up to 5 milliseconds. This guarantees that the downstream neuron will be ready. It also guarantees that this is going to be another way to guarantee it's a one-way process, that we only go one direction. So this is the rate limiting step of nerve transmission here. We have that built-in delay. We want to make sure that the downstream neuron is ready, because neuron fires and fires and fires again and again and again. We want to make sure every time the presynaptic neuron fires, the postsynaptic neuron's ready. And the synaptic delay guarantees that for us. On the postsynaptic side, one of two things can happen. We're going to generate a graded potential when we have the neurotransmitter landing on the postsynaptic side. One of two things will happen. It depends on how much neurotransmitter gets released and how long does it stay there. We can create a membrane potential on the downstream neuron that is what we call it's either going to be an EPSP, an excitatory postsynaptic membrane potential, or an inhibitory postsynaptic potential. In other words, we're either going to generate an action potential or we're going to drive us further away from generating the action potential. If it's inhibitory, we're going to change the charge in the downstream membrane further away from threshold. If we're excitatory, we're going to end up generating a graded potential that will lead us to threshold, maybe. So, okay, this is a great place to stop because I don't have enough time to really go into this. So we'll stop here. And anybody have any questions? Okay, then I'm going to get us out of here. We'll wrap up this chapter on Monday and move into the next chapter, which is called Chapter 12. It's on the brain and spinal cord. So let's get us out of here. And I will see you all next week.